Hello, everyone. My name is Alex Iltz. I'm the producer of A Petri Dish, which is brought to you by the College of Biological Sciences in collaboration with the Bell Museum. Thank you so much for joining us this, I would normally say evening, afternoon. For those of you that haven't attended A Petri Dish in the past, these conversations are normally held in person in an informal setting where we explore topics related to how biology affects our lives and what it means for the future as well. We moved online along with everything else, but hopefully with the advent of vaccines, we'll be able to return to a more in-person and analog style of meeting, if you will. So just a few quick announcements. If you like this Petri Dish, please come back again virtually next month for a discussion that addresses racism and equity in STEM. You can also join the Bell Museum for a virtual star party to celebrate the landing of the Perseverance Mars rover this Friday. And next month, catch a conversation about environmental justice challenges in the Twin Cities in conjunction with the Climates of Inequity Stories of Environmental Justice exhibit. More information can be found at bellmuseum.umn.edu. And this afternoon, we have 3 university researchers here for what promises to be a fascinating conversation about vaccines in general and what that may mean for combating diseases like COVID in the future, now and in the future, really. So I apologize, but looking up here, I'm getting dripped on. This isn't a virtual background. This is, I'm really surrounded by palm trees and it's lovely. So we typically kick things off a little bit of trivia. We're going to do that online as well. And so I'd like to invite Claire Wilson to lead us in some trivia related to today's topic. Claire is a science communicator who translates complex research for a general audience. So Claire, thank you for joining us.
Thanks for having me, Alex. And I now invite all of our panelists and Dean Forbes to kind of join in. They will be your cheerleaders as we embark on trivia together.
Forgot my pom-poms.
Okay, so here is the first trivia question. The first vaccine is often credited to Edward Jenner, who famously used material from cowpox pustules, a very nice word, to provide protection against smallpox in 1796. There's evidence that Chinese experimented with smallpox inoculation earlier. What year? And all these are in the Common Era.
Do all the questions involve the word pustules?
You know, I was really going for that, Alex, but I couldn't quite squeeze it in.
All right, well, better luck next time.
I don't want to rule it out for future Petri dishes, though.
Good, good.
Nothing,
All right, we'll give it
nothing worse than squeezed pustules.
nothing worse than squeezed pustules.
Power for the win.
I'm glad we made some progressions with our vaccine development.
It's always important that pox come from farm animals like cow and chicken.
What's a small on a farm?
Well, that's a deeper subject.
Oh, all right.
So we could banter all day. I'll give it another 2 seconds before I close this poll out. Okay, final answers in. So here's where we landed. The correct answer is in 1000 Common Era. So much earlier. I couldn't find too many details of what exactly they did and how successful it was, but there's some record that was that early on that they were experimenting. All right, second question. What is the key difference between mRNA vaccines and more traditional vaccines?
You're gonna steal all my questions for later.
Not too many. I tried to just give a little teaser, but we'll see. I hope I don't make your life too hard, Alex.
Oh, that's all right. My life's not so bad.
You do have more greenery than all of us. Okay, we seem to be in pretty good consensus here, so I'm going to end it in another second or two. Okay. So a very strong consensus on the final. mRNA vaccines do not use an inactivated or live virus, whereas traditional vaccines do. And that is correct. Going on to our third trivia question, plowing right along.
Audience is on top of it.
And I sure are. What COVID-19 vaccines leverage mRNA? And you can pick multiple, you should be able to pick multiple answers here.
That was a clue, I think. Like,
This might be.
You should have been my organic chemistry professor in college. I would have done much better. Here's some clues for the answer.
Don't sweat it too much. Okay, we're gonna go ahead and close it out. Looks like people are mostly done here. Again, there's pretty strong consensus with Pfizer Biotech and Moderna vaccines, and that is correct. So those two vaccines are leveraging mRNA technology, whereas Johnson & Johnson is not. All right, 4 out of 5. What is the most accurate analogy to describe messenger mRNA's role in a cell?
I want to see if they land this one. I am definitely going to be slacking for all of them. It's
And hopefully all of our panelists are in consensus about the answer, but feel free to debate me about the wording on this. I am not a faculty member who teaches this, so.
I'm thinking if they nail this one, that maybe we ought to pick some of the audience to be the panelists.
All right, we'll give it another couple seconds here. All right, I want to make sure all my panelists agree here. So what do you three think?
oh, on the spot.
The hardest question, maybe. And you could say no answer is correct. I won't fight you on that.
Can you paste them in the chat?
Oh yeah, someone just wrote in the chat. I can vouch Professor Engelhardt.
No. I was saying, could you paste the answers in the chat so we can look at them while we talk?
Oh yeah, sorry, I didn't share the. Do you see him now?
Yep.
Okay. Someone said you taught him this, so.
Like every class, I feel like there's been a COVID angle to everything this semester.
So do we agree? Are we?
Yeah, where do we agree? Is it so I thought that it was number 2. mRNA decodes the blueprint DNA and delivers it to the factory floor. But I might be incorrect here. So I defer to you all.
I felt like
bad analogies.
No, I honestly felt like you could make the case for all of these being right, because really mRNA, yes, there's that DNA blueprint, and then obviously the ribosome really is the factory floor. So it's delivering that. And in a way, it's decoding it because the DNA gets transcribed into RNA. But there also is this controls production element. That was actually the one that I picked, that third answer, that
As did
is an
I.
important part of it.
I came up with a really bad question, so I'm glad I'm not a panelist in this Petri dish.
Hey, we bring the best panelists. See, this is great. It's evidence of that.
No, I thought it's a great question. No wrong answers here, I really feel like.
Well, that's good. There will be a lively discussion then. And I'm looking forward to learning more. Okay, hopefully the last question will be a little bit easier here. What statement is true about mRNA vaccines?
I noticed that you didn't put in there that it's secretly the government putting a microchip.
That would have been.
No, I was going to ask a
right?
question about common misconceptions, but I thought that would go down a really dark road.
I didn't think that was the government. I thought that was Bill Gates, but go figure.
Oh, I don't know the actual thing that people are saying. Sorry. I was just making it up on the fly.
You're obviously not following the right social media posts.
I'm not on social media. Yeah.
Oh, and I'm realizing that this is a very poorly worded question. I forgot to add my not in there. So apologies to all of our attendees out there. Again, the last 2 questions, there's no correct answer. But I did make you think, which is good. So what statement is true about mRNA vaccines? They're actually all false. So there are no true answers here. My apologies. That one was supposed to be do not, but these are all false statements.
Well, they remain stable at room temperature for a few minutes.
For a little bit. That's true. That's true, Dean Forbes. Yeah, for a little bit. Well, thanks for your patience. This is the first time I've done trivia for Petri Dish, and I don't know, given that performance, whether I'll be invited back. But hopefully you had some fun with thinking about some of these questions.
Well, thank you, Claire. I enjoyed it for sure. Well, I think it also tested our panelists, so that was fun too. I will now give the floor over to Dean Valerie Forbes, who is the Dean of the College of Biological Sciences, and you are our winner of the pustule jokes. So take it away.
The highlight of my day. Welcome everybody to the College of Biological Sciences Petri Dish. As some of you know, this is one way that we try to share our science with the broader community. And we're very happy that you've decided to enter our virtual Petri Dish and be part of our latest experiment. And I would encourage you to be active participants. So for these conversations, we bring together people from our college, the university, and beyond who have expertise and a point of view to share around topics of broad interest that also have a biological bent. And biology has certainly been front and center throughout this pandemic. The race to develop vaccines has given us all a front row seat to science happening in real time. And one of the most interesting twists to the story is the emergence of this mRNA-based, these mRNA-based vaccines. And as we've just learned through trivia, the two vaccines currently approved in the US, produced by Moderna and Pfizer, are based on this new mRNA technology. And they're the first vaccines of their kind, and as such, are really unfamiliar terrain for the science curious. So even as we begin to hopefully bring this virus under control as vaccines reach more and more people, scientists are still hard at work investigating the origin of COVID-19 and how it impacts human health. The science underpinning these vaccines is truly groundbreaking, and as our panelists will no doubt tell you, the potential to use this approach for not only vaccines, but a range of other applications is wide open. So with that, I hope you'll join in with your own questions and comments, and I want to thank you for being here and hope that you enjoy the discussion.
Thank you so much. So we're going to move on to our main program here. Before we get started, I want to encourage you all to submit questions through the chat function. So Claire, who you have already met, is going to sort of aggregate those, and we'll be checking in with her as opposed to what we normally do in our live events. We'll be checking in with her a few times throughout the program to make sure that we're getting your questions answered. And she's going to try to ask the questions that are being asked most frequently. So definitely, if you have any questions, send those in through the chat. We have turned on closed captioning. So for those of you that want that, that's great. If you don't want that and it is driving you nuts or is going across my face, which might be a good thing, you can turn it off though by clicking on the live transcript button on the bottom bar. We're going to record this webinar and it will be posted online, and we'll also be sharing the link of the recording. So enough tech announcements. I will be asking our guests, I'll be introducing our guests and asking them to talk a little bit about what they do and what their backgrounds are. And as always, I like to, probably just for my own entertainment, ask them all a silly sort of ridiculous question. And in this case, I want to know from each of them just what is the absolute weirdest thing that they know about viruses? Because they already seem weird to me. Are they alive, not alive? It's a whole thing. And I know so little on this topic. So I'm really excited to have them here and to get to learn a little bit about what's going on with this. With that, I would like to introduce Dr. Erin Engelhardt. an assistant professor of genetics, cell biology, and development. Erin studies ways to use DNA and RNA in ways beyond their well-known role in biology as information carriers. So welcome, Erin. You want to tell me a little, in a way I can understand it. Clearly the audience is you can give them all the details, but in a way I can understand. Can you tell me a little bit about what you do?
Yeah, thanks for having me. It's great to be here. And so yeah, our lab does a lot of work with RNA and especially RNA tags that let us visualize RNA that we might do or that we've historically been able to do with proteins, but haven't really been able to do with RNA. And so there are RNAs that can generate fluorescence that we use in a number of roles in the lab. We can use these for cellular imaging. In the last year, like a lot of labs, we've kind of been focusing on COVID, and we have some work using these in COVID diagnostics.
Excellent. That's very cool. So when you say fluorescence, does this mean you can make like my DNA glow? Is that or RNA glow?
We literally can. You can we can take a tube of RNA and you'll get a green or red or orange glowing tube.
That's awesome. So what is the absolute weirdest thing that either you know about viruses or you want people to know about viruses?
So I'm going to let the audience in on a secret. We had this in advance, so I, did a little bit of background work on this. So I was going to come and state with utter confidence that the weirdest thing I knew about viruses is that there's a virus called Mrna virus, and that's the largest virus, and it's almost as big as a microbe. It's a couple hundred nanometers across. Bacteria, are on the order of 1,000 nanometers across. And I went to look because I wanted to get that number to say about how large it was and how far across it was. And then I looked, and it turns out in 2013, they discovered a bigger virus called a Pandora virus. And I thought, okay, well, that's the biggest virus. But then I found out that in 2014, they discovered an even bigger virus called a Pitha virus. And that's actually bigger than a micron across. So the weirdest fact about viruses that I know is actually one that I learned this morning when I was preparing.
Glorious. So you've already gained something by being here.
Yep.
That's wonderful. Up next, we have Dr. Kathleen Boris Lowry, who's been a faculty member since 1996, initially at Ohio State and at the U since 2015. She's trained many students who are scientists and professors in the USA and around the world. She says that student success and scientific discoveries are the joys of life as a professor. That is inspiring. I love it. Thank you so much for being here. Oh, I think you are muted currently.
There we go. There we go. Yeah. Pleased to be here, Alex and all. Thank you for the invitation.
Oh, absolutely. So can you tell us a little bit about what your research is?
I'd love to.
Excellent.
So I've studied viruses for a while, and it turns out that Claire, on her trivia, the question that all three of us were debating about what is the role of RNA, that's along the lines of what I do. And the bottom, so I picked the bottom one because that's what I do. And RNA not only carries the information from DNA into to hook up with the ribosome, which is the factory to make proteins, it actually can control how well those proteins get made. And so viruses have figured out ways to manipulate that process that really teach us about what kind of activities the cell can do to fight off viruses and to create diversity in how they respond to stress. And so that's what I study, is RNA structure related to how well proteins get made. And we use viruses as little microcosms that we can manipulate to understand how the cells work.
So you're using the viruses for your own ends.
We're trying, yes, you could say it that way. You could also say that we are trying to
diabolical evil scientist way.
Well, it sounds like, that's a very powerful statement, but actually viruses are so diverse and do so many unbelievable things, which is a part of your next question, that we're trying to figure out what these little teeny viruses do. So we're the big guys, right? But they're the tricksters. And so we're trying to decode that. And by doing that, we learn about how the cells work. And, not just a normal cell. We want to know about cells that are sick, cells that are healthy, cells that are metabolizing, so we can understand how to make people healthier and animals healthier. And so they're really, we're following their lead, actually.
Nice. So what then is the weirdest thing about all of these diverse, crazy viruses?
Well, so like Erin, I had this question too in advance. And so I just had to Google up some things. And so, just how many viruses are there? Well, there's this great analogy in National Geographic that there are as many viruses as there are stars. In fact, there's 100 million times per star as many viruses. So, whoa, there are a lot. And actually, like 2/3 of all human pathogens are thought to be viruses. So bacteria, yeah, for those who study that, or fungi, or if you have ringworm, that's a fungus. Well, those fungus don't get many chances to cause disease. So I think it's really cool that there's so many viruses, and just like differences in size, they've just, not only are they there, they've really affected us. And so so I study these retroviruses. They're retro, like fashion or whatever we can talk about.
Good.
And they integrate into our DNA. And they change our DNA. And you'd say, oh, that's terrible. It's going to cause cancer. It's going to cause AIDS. Well, once in a while it does. But actually, when you look back over evolutionary time, we have like 8%, 10% of our DNA has retrovirus hallmarks. And they are actually jumping around. And it's thought that our brain development, the plasticity of our brain, is due to the diversity added by these viruses.
So I want to cut you off there. One, for time, that is an amazing answer. Two, because I do want to actually talk a little bit about some of how that has been wrapped into the understanding or misunderstanding of our current situation. So
okay.
You're way ahead of me. You're all way ahead of me, but I wanted to, hold back for a minute for me. And finally, I want to welcome Dr. Jason Baran, an assistant professor of pharmacy. He also practiced as a community pharmacist for 22 years and has served as the president of the Minnesota Pharmacists Association. So thank you for being here, Jason. And I think you're also muted at the moment as well.
Sorry about that. So yes, I'm very happy to be here with this this fabulous group of people, fun and intelligent at the same time, which doesn't always work out that way. I'm less of a hardcore researcher. The research that I've done and the studies I've been involved with are delivering pharmacy and health services to the communities, and the best way of doing that. Since I've been at the college for the last 5 to 6 years, my interest has really turned to educating the public in a way that they can accept and is comfortable to them, and to help health providers provide the information in the terms that our communities can relate to them and accept them. Regarding the virus, the thing that just gets me, that I can't get by, is that they're really not alive, but they're really not dead either, right? Because they don't convert, they don't change food into energy or anything, but yet they evolve and they reproduce. So that's pretty cool and pretty scary at the same time.
Agreed, especially after learning how many of them are out there. I'm glad I don't have a runny nose like right now. So what is the weirdest thing that you know about viruses? And then I'll have all the panelists after this turn on their mics just all the time.
Well, I was going to use that as my weird thing about viruses, is that they're not really a living, they're not alive or dead, but we'll stick with that for the interest of brevity.
That's legitimate too. Well, thank you again all for joining us today. I'm really excited to learn about this. Obviously, it's very timely. So vaccines, given that I have a panel of experts to talk to here, I guess the first thing that I want to cover, which is probably too basic for many of our audience members, but I want to just make sure that I have the basis down for this. What exactly does a vaccine do? Why does it make us immune to a disease? So I will throw that out to anyone who would like to answer it, and I'm going to get Jason and Kathleen to turn their mics on, so you guys can feel free to chime in whenever. I don't know. Does anybody want to take my most basic of questions?
I can give it a shot. Yeah. So what do vaccines do? They pretend. They pretend that they are the microbe that we don't want. So they look like it, but they look like one aspect of it, but they don't have all the activities. So I just heard a good analogy today. It's like a car that you take out the engine, you take out the seats, looks like a car. The immune system goes, whoa, that's a car. I'm gonna like do something to prevent it. I'm gonna react to this car. So this could be any sort of molecule. React to this car. And so the immune system responds and gives this protective effect, but the car can't go anywhere and cause disease. So that's an analogy. Maybe Erin or Jason want to add to that.
I think that was pretty good. I think that I once bought a car without looking on the inside, so I can relate to that. But yeah, it's.
And now you'll never buy a car again. Is that it?
Not without looking inside and taking it for a test drive. Yeah, just it's kind of tricking the body, and we want it to trick the body, right? Because we don't. We either want to trick it or we want to give it a very mild, mellow infection that our body can easily overthrow and adjust to.
Yeah, I would definitely concur with the other panelists. I think that one other thing that comes up with this too is that there's this idea of innate versus adaptive immunity in your body's immune system. And, this innate immunity is kind of alarm bells that go off when there's an invader, and then the adaptive immunity is kind of where your immune system learns to respond to something. So it's sort of like what Kathleen mentioned about, you're kind of training, you're tricking your immune system. And that's something that gets balanced in these vaccines, where you kind of want to have these turned on in exactly the right way.
So I love the idea of them, pretending or acting in there as a fake virus. It seems very microscopically Shakespearean to me. So the innate versus acquired immunity, did I do that right? So these are all stimulating the acquired immunity, and presumably we don't have issues with things we have innate immunity to. Correct? No?
So the innate immunity is kind of a non-specific thing that gets turned on when there's something foreign that comes in. And so like that's part of a response to a vaccine, where when you get the vaccine, you need that turned on because it recruits the adaptive stuff that is actually going to be involved in the response.
Okay, excellent. So now, as Claire gave away in the trivia, some idea of vaccines, and vaccines in general have been around for a while. There was something with cowpox and all of these kinds of things. But the vaccines that we're talking about in terms of COVID, these mRNA vaccines, this is new, right? And so what is it that is so new about these things? What is it that is different? And I guess my real interest is, why is this so groundbreaking? Why is this something that is such a focus of how these vaccines either work or are produced? From anyone, really.
Well, I could say that the technology has been here for a while. I know they did some early work in 2002 and 2012 with MERS and SARS to try and get some of this technology down. And I think it was around before then. Maybe my colleagues can correct me on that or clarify. But I think the real groundbreaking thing was to mobilize that technology in short order and adjust the other processes around production around it. But I will let the other two pipe in on the technology aspect of why it's groundbreaking.
Well, I would just say that
I thought that the tech was new. So yeah, I would love to hear more about the technology and what aspects are groundbreaking. Sorry, Kathleen, I didn't mean to interrupt.
Oh yeah, I was just going to say that, you know, some of these other vaccines, the traditional vaccines, look like a virus, but they might have a piece missing. So it's not going to replicate and cause disease, called attenuated, like the polio virus vaccine is it's an attenuated virus. This is, and so when you put that in people or animals, it comes in, it has to be in a solution that can, in a complex solution. And so some of the anti-vaxxers, which you might get to, have, you know, people say, oh, that stuff you mix with it is bad, it's poison. And so, you know, there's others who say that is totally not true. Well, the thing with the mRNA vaccine is that it's not in any of that extra material. It's just in a little bit of salt water. And so, and it's just one little piece of this RNA that gets into cells, and the cell machinery uses it to make that viral protein. And then poof, it's gone. It only lasts for a couple days tops. So gee whiz, it seems like it could be a lot safer, or a lot, let's just be conservative and say less unsafe than a live attenuated, or than an attenuated.
I guess my first thought when you said live attenuated, and then, you know, explains to me what that was, is if you have removed some gene, some aspect of the coding region, you know, my first thought was, could it mutate randomly and become active again when it's being used? And did that happen ever? This is a bit of an aside, but I'm curious if that is a danger with those, and maybe something that is also safer about this mRNA component.
Yeah, with oral polio vaccine, what Kathleen mentioned, you know, when it's an attenuated virus, there is some level of risk that the virus, you know, can replicate and go on and infect people, and that's simply not possible with the mRNA vaccines. You know, as she mentioned, this is a piece of RNA, just like your cell would make in the course of making protein, that codes for a region of the viral genome. And the way that both of these mRNA vaccines work is they code for a protein that is on the surface of SARS-CoV-2 that it uses to get into your cells. And so that's kind of the training wheels, or the trick that Kathleen mentioned, to get your cells to recognize that and start raising antibodies to it. And then kind of the other thing that goes into it that was sort of the trick that we had to get the hang of in order to actually reduce these to something where we're using them as vaccines was packaging them. You know, Kathleen alluded to the idea of an attenuated virus. These mRNA vaccines come with RNA, but it's also complexed with kind of an oil. There's what they call a cationic lipid that makes this complex with RNA. And that's both what lets it get into the cells and, you know, do its job. But that also kind of ties into this innate immune system thing that I mentioned, that the modulating this immune response. These lipids, kind of this fatty packaging that we use in the vaccines, that's involved in modulating the immune response to the mRNA too.
So there's like saline and some lipids in terms of what's in the shot. And I guess how did we know this was going to work? I mean, is this the Jason, what you were talking about, that there was a bunch of buildup and sort of the basic science on that, where people were looking at these aspects and then they were pretty confident if they went down that road in this time frame, they might be able to get a vaccine out of that?
So that's one of the things that has really helped. Coining warp speed, Operation Warp Speed, or anything really is a name for it. But the reason it really worked fast because of the existing technology, our experience with vaccinations and immunizations over years and years with a large variety of of vaccines and understanding how they work and why they work. And the RNA messenger RNA works by what they do is they identify a specific part of the virus. So if a virus, think about this, if you get infected with a virus, in most instances, it's going into the cell and it's taking over, it's turning into a vaccine factory. It's messing with the DNA, it's taking over all the factory machines, if it were. Whereas with messenger RNA, they've figured out what the code is that the virus uses to create a particular subunit or a particular part of protein, in this case, the spikes or the parts that make the corona. And the messenger RNA gets into the cell. It does not enter the nucleus. It interacts with the ribosomes and the machinery to some extent the same as the virus does, but it doesn't mess with the nucleus. And it's only coded to create that one protein that makes the spike protein. So if you visualize the virus and the messenger RNA way that they work, I'm going to go with the messenger RNA rather than with the virus.
So I do want to break for audience questions after this in just a minute here, because I also want to get into sort of the rollout of this and the concerns people have and some of the bigger sort of societal questions with it as well, of course. But what you're telling me in terms of this, and I'm really trying to wrap my mind around how this thing is working and how it differs. But it reminds me a little bit, and you can correct me with how wrong I am. It reminds me a little bit of the practice of putting out sterile mosquitoes into a population where they try to mate and breed and they can't. And so it kind of works against the organism that you're trying to defeat with some of its own machinery, right? And so in this case, you are trying to get it's a fatty lipid to deliver just the piece that you want to get the immune system to recognize it. Am I at all close?
No, I think you've touched on a lot of sort of, we talked at the beginning about, our virus is alive and what goes into that. And, there's kind of this continuum of life where you have something like a bacterium or human that's, this thing that can self-replicate, make a copy of itself, and a virus that needs sort of that host organism to replicate. Then kind of going down that continuum, you would have these mRNA vaccines where they can't replicate. And so that's, going in, like Kathleen mentioned, it hangs around for a little while and does its job, but you're not going to get, more copies of it.
Yeah, and I guess that was my thought with the analogy to the sterile mosquitoes, is the sterile mosquitoes aren't going to make more of them specifically. And so they hang around for a while, mess with the system so that things don't work quite the way the regular mosquitoes, in this case, the regular virus, was attempting to do things. Excellent. Okay, so I feel like I've got a little handle on that. I wonder, Claire, if there are some audience questions that are clearly more intelligent than mine.
There are a lot of audience questions. I think from all the Petri dishes we've done, this is the most so far, so gear up. A lot of them relate to more the safety and more long-term pieces, so I'm going to save a lot of those questions till later.
Yeah, I think we're going to move into some of that next.
Yeah, but I'll ask just a couple of, maybe two quick ones. So someone who asked this used interstitial spaces. So I think whoever asked this is a scientist out there. So they asked, once the mRNA causes our cells to produce the spike proteins, what happens next? Do our cells just spit out the loose proteins into the interstitial spaces between cells? Which you'll have to define that for me.
That's a good question. Yeah, what becomes of it, I guess, is really, that's how I would ask it without knowing all the other things.
Well, I would just chime in and say, if you think of a cell of like a balloon, and the RNA goes in the balloon and the ribosomes use it to make the spike protein, the spike protein has a ability to go to the edge of the balloon and want to pop out. Okay. And the immune system is supposed to be really fast and slick so that before it even pops out, the immune system starts to react to it. And if it does pop out, then, well, my colleagues can talk about that more, but I would just say that it's not like it's pushing out tons of the protein, that the protein starts doing its work even before it gets out. And when it gets out, what is an interstitial space? That just means space between the cells.
Thank you.
And your immune system is constantly going around looking for things that don't belong, right? And when this spike protein is presented and they figure this out, then they can start the immune process, which takes some time. It doesn't happen immediately. That's why it's so important to train ourselves in this manner before we get the full strength COVID virus, because it takes a while for our bodies to learn how to fight that, whether it's the spike protein that's attached to the live virus. And then while our body is mounting an immune response and learning about it, this virus is replicating and going to cell after cell after cell after cell, wreaking havoc on all of these cells, which is where the damage occurs.
Yeah, I would agree with Jason, Kathleen. And I think that this kind of touches on the question you asked earlier about, you know, how did we get here and what went into this? And a big problem with all therapeutics that involve nucleic acids, whether it's mRNA, whether it's DNA, you know, we've been working on this for decades, and delivery kind of goes into this and exactly how you get it into the cell and in the place that you want. And we know from these mRNA vaccines that you know, you're getting the shot in your shoulder, but a lot of it actually goes to your liver. And when you inject nucleic acid, a lot of it ends up there. Some hangs around your muscle, and that's getting translated as well. But you know, getting these to the right place has been something that's been studied for, like I said, decades in a number of contexts, both in mRNA as well as other nucleic acids, so DNA and RNA-based therapeutics, and that kind of goes into it. But where it's expressed, and you know, whether it's the liver or your muscle, your immune system, as Jason and Kathleen said, sees it.
Excellent.
Great.
Do we have another?
I'll ask just one more. So a couple people had questions about how mRNA vaccines kind of differ from traditional vaccines. And one of those questions is, do traditional vaccines interact with the nucleus of the cell?
It's a good question.
I think people worry about that because the idea is that it would go in and permanently change the DNA of the cell. And these mRNA vaccines are not thought to go into the nucleus, and they certainly are not thought to change the DNA. In terms of, well, other, you know, the live attenuated, I don't think that those would go into the nucleus. What do you guys think? Erin? Jason?
I mean, I think if anything, it's a lower risk with mRNA than other technologies, because, you know, like the viruses that Kathleen works on, these retroviruses, they have an enzyme called reverse transcriptase that does something where, you know, if you look at your, I have a lot of students from my class here today, so everyone's going to remember, they always come back to the central dogma at the beginning of class. You know, you have DNA goes to RNA goes to protein.
Yeah.
RNA can get copied into DNA, but that's not typically something that's happening in biology, and it's kind of an edge special case. So, you know, for you to have genome incorporation, that would have to happen, and that's not usually happening in a healthy cell.
Great. Well, I'll be back, I'm sure, with a lot more questions.
Excellent. Thanks, Claire. And thanks to the audience too. Those were fantastic. You know, you had mentioned, Kathleen, in response to that last audience question, that people were concerned about things. And certainly there has been, it seems to me, a lot of buildup of concern towards vaccines in the past decade, even before before COVID hit. And now we have new vaccines based on a new technology. And so I'm wondering both how we know that those things are safe, and then also, as kind of a two-part to that, is how did the regulatory agencies know that they were safe enough to roll out? What is that process?
I could start that one off.
Right.
You know, as I mentioned, the technology existed. It was putting it to practice, which they did. And again, I think part of one of my interests in interacting with the communities is vaccine hesitancy. And that's where my interest in learning about this is because I can't expect people to understand why they should do something if I can't explain the details in ways that they can understand, or ways that I can understand, probably more importantly. But again, what throws a lot of people off is the warp speed, or how, you hear that it takes an average of, I believe, 10 years for a vaccine normally to come to market. But the reason this happened is because, number one, we had the technology already. A group of experts, including not just the researchers at FDA, CDC, NIH, review all of the information throughout the trials. Now, they have to go through the same process of clinical trials that you would do for any new drug or vaccine. When there's an emergency declaration, it opens up the opportunity for the emergency use authorization. And all that means is, what you try to do is, rather than do one stage now and next stage and the next stage, which is the phase 1 trial, trials start with just a few people to make sure it's safe. It's not really causing major problems. If that goes well, then it goes out to larger numbers, hundreds or thousands of people. And they're constantly checking for safety throughout this, but they're also looking for efficacy, as well as other aspects. And then the phase 3, which is usually the last phase before it comes to, it either gets shot down or approved, it's to thousands and thousands of people to make sure, again, it's safety, efficacy, and effective, and does what it's supposed to do. In an emergency use authorization, what they do is they go into phase 3, where they're doing thousands of people, but they get to a point where they can observe the data that's come out of it and decide if the benefit that, in this case, the country will get and the world will get in their own emergency use authorizations, if they choose to use them, really outweighs any potential danger from what comes out of using the vaccine. And a lot of it goes into not just the physical harm, but the loss in life, of course, but the loss in what it's doing to our economy. What's it doing to, what are the other effects it's having? But even if it were just for safety reasons, they would have approved it. So, they would have authorized it. So these vaccines are authorized right now. They have not been approved by the FDA, but as we go through this process, they're continuing those phase 3, and there's phase 4 too after approval, that they'll monitor for safety as well. But you should feel totally safe about how they've done this, and the fact that we're saying we're going to do this as fast as possible. Shortcuts were not taken on up to that point where they decided that the safety is there.
I'd like to add to that.
Please.
Because the point is, that there's a robust safety process. And when we think about the old-fashioned vaccines, which I'm sure we'll still be using those at times, we have to, for the polio virus, we have to figure out which gene to take out, so that and then, we have to test all these different viruses, take out one gene and figure out which ones are no longer pathogenic. Then we have to scale it up and grow it in eggs. And then we have to make sure that there's no contamination. There's all these steps. But look at this. This is the RNA that encodes just the spike protein. And we don't have to make the protein. We don't have to quality control the protein. We let the cell do all that work. So when that stack of paper of the, for the approval, instead of being 10 miles long, maybe it was 1 mile long, just because there are fewer things to look at. So it's very much a simpler vaccine. And so I think that's, when we think about warp speed, it's maybe if we think about it on a per nucleotide basis or per gene basis, it's not such a warp. speed. Maybe it was just, doing a good pace.
Efficient.
This is so much simpler than a conventional vaccine.
Yeah, there's a comment in the chat where the major shortcut taken was to start manufacturing at commercial scale prior to EUA, and that really touches on exactly what warp speed was about. Warp speed wasn't about going faster; it was just about deciding to spend the money, because we're losing so much money every day. This comes up all the time in science, where there's more meritorious science than we have to fund it. We just made the decision collectively as a society that whatever this cost, we were going to pay for it. And looking at it, it's been a very successful bet. When you look at what Jason mentioned about these, phase 1, 2, 3 trials, you try and design a study cohort of people where you have a, statistical insight into a population. I think that went a little bit faster than we might have even expected, because there was just so much COVID circulating in the US in particular, but in the world. And so we kind of got the answers a little faster. We're now seeing, when we look at kind of more messy population level studies, the best example being Israel, which has some of the highest vaccination rates, that we're getting pretty much the same results there. It's effective, and it's effective against some of these new variants that we're seeing come up. Israel, the predominant form of COVID that's circulating is, you might have heard B117 mentioned in the news. That's the main one circulating in Israel. It's effective against that. You should be confident in these vaccines. They're, very safe, very effective. And as Kathleen mentioned, it's a much simpler process. We're not doing something where we're trying to make a virus that can sort of infect someone but is not going to really take off. The way that these are made is we take synthetic DNA, we use a purified enzyme, and that in turn makes RNA, and then they get packaged into these lipid particles. So it's very controlled in a way that something like an attenuated virus vaccine is not.
So in terms of safety, I think that's great. And you're also saying that it's showing to be very effective. That made me think about, is getting a vaccine, this vaccine, say, or these vaccines, more effective in terms of long-term immunity than having had COVID? Because certainly in the news, you hear about people who had COVID and they don't have antibodies for it now. And maybe that's a two-part thing. Like, could we do a sample of my blood and figure out every virus I've ever been exposed to by doing a full suite of the antibodies that you find? Or, I guess I'm trying to understand, if you can get the disease more than once, how is a vaccine going to prevent you from doing so?
I think a lot of this is kind of a question of large numbers. If you look at, the world population that's had COVID, if I was talking with a colleague about this, and we made the comment that, if you feed 100 million people a hamburger, you're going to see some weird stuff happen, even though, hamburgers aren't inherently dangerous. And there are going to be people that obviously get reinfected with COVID, but it does look like, immunity is durable. It lasts quite a while. The antibody question comes up, and those do fade over time. Something happens when you get the 2 shots, where they call it a prime and a booster dose, where you start making antibodies, and then the second dose, you start making better antibodies. But then beyond that antibody-based immunity, there are cells in the immune system called T cells that are also involved in immunity. And that's kind of the durable, longer lasting immunity. And there's been less work done on that, just because they're harder questions to ask. There's been some limited work in Sweden. There was a study of just about 200 people looking at this T cell versus antibody based immunity. And there's actually more people with the T cell based immunity than antibody immunity in that study that they found there. And it's a tricky thing to interrogate, because looking if you have antibodies is relatively easy. You can have kind of a pregnancy test type thing where you see a line show up on a strip. The T cell question is harder to ask, because that involves experiments that experts are doing in a lab.
Okay.
And I would add to that, and one of the questions on here is that the safety, science is not perfect. Anybody who's followed science, it's not perfect. It's a learning process. So the best we can do is take the knowledge that we have from the past and the current knowledge that we have, and try to predict how that's going to react in the future. Now, we don't know the long-term protection because we've not used these vaccines before, right, to any large scale. And the reason that we don't know that we could still shed, so this is my vaccine card, and if you look, my second dose is going to be tomorrow. So I will tell you, I like to walk the walk as well as talk the talk, but I'm looking forward to it because, and I assure you though, that after I get this, I'm still going to be practicing social distancing and wearing my mask, if nothing else, to protect other people, because I don't know if I'm still shedding or if I can still carry the virus. But it really, like I said, you have to understand there are 385 million people that have died from this virus. That is something that we know. We don't need to speculate about that. So my request is for the person who said, how do you talk to the public? Explain, some of the things that we've talked about, and that the level of trust. And that's another thing, is the government and these manufacturers are not going to release these vaccines if there is any chance that there might be a major problem with, because what that's going to do, that's going to reduce the trust even further and make it much more difficult to handle these pandemics.
So I see that there's a number of questions popping up about a variety of broader topics. I know we're closing in on time here, but I wanted to ask another one in terms of durability and thinking about that before we address some of those questions about some of the broader societal things with the audience questions that I see they're astutely asking. But with the variants that are coming up, is that concerning? What aspects about that might be concerning? And is this something that if a variant comes up that releases it from the vaccine, does it bring us back to square one, or is this going to be easier to respond to in the future?
I'd like to take a crack at that with my mouse. Okay. So
I'm all in.
talk about props. Okay. So when we get the RNA, we make the protein, and then the protein is responded to by the immune system that makes antibodies. Let's just focus on antibodies for this example. And so that the protein looks like this, and the antibodies stick all over it, and they kind of they stick on the button, and they stick on the left side and the right side and the back side and everything. So now you get a variant. Well, it still needs to be a mouse. It still needs to be a virus that can get in a cell. So it can't be totally changed into a banana. Okay. But it's now going to have a little, maybe this wheel piece is now a different shape. Well, so that means that the antibody that's stuck there maybe isn't going to stick on so well. It's going to be falling off a little bit. But what about the ones that are stuck all over the other places? There's still a lot of antibodies, a lot of specificity to that protein. So that's how I think about it. I really like to hear how my colleagues think about it, because there's so much in the news about this.
Yeah, I completely agree. I mean, to add to what Kathleen said, I think that, it's important to remember that we've eradicated diseases before. We've eradicated smallpox. There's no polio in most of the world. And this all happened while these diseases were under vaccine pressure, that they had the opportunity to evolve and do vaccine escape. A lot of what we're able to do here, and the reason that we're seeing these variants come up, is because we're able to ask questions that we weren't able to ask before. The fact that we had, the reason we got these vaccines so quickly was in part due to the fact that it's now almost a trivial thing to do to get the sequence of a given organism. And that's what enabled us to produce some of these. And so kind of going into this, and, with some of what Kathleen mentioned about the simplicity of production, even if there is issues with vaccine escape, which I'm a little bit more sanguine about than I feel like you read in some of the news stories, these are quicker to patch than you might have with another production method. Because like I mentioned, there's this workflow where you have synthetic DNA gets made into RNA with a single enzyme, and that gets packaged into these nanoparticles, and that's your vaccine. it's much easier to adjust something like that than engineering a new virus that's going to be what goes into your attenuated virus vaccine, for example.
And I'll just add a quick point to that, so you can think about this, is that things evolve over generations, right? It doesn't just happen on a single individual. I'm not going to evolve into anything. So with that in mind, one of the biggest things that we can do as a society to reduce the chance of variants is to knock this thing out, the sooner the better. Do the social distancing, wear the mask, get your vaccination, because the longer this stretches out, the more chance there is for variants in the future.
That seems like a great note to move over to audience questions. That was an excellent point.
All right, I'm gonna I'll grab a couple. We have over 40 questions that have come through. So
don't be offended if she doesn't get to your question.
Exactly. There are a lot of good ones. And I encourage the panelists and Alex, if there's something that you have seen that you are just really want to answer, feel free to interrupt me. So I'll pick one, but take a second, kind of look through. And if there's something, Alex, that you really want to address,
all right,
I will follow your lead. So one question that I would be remiss if I didn't ask is, we've talked a lot about how different mRNA vaccines are from traditional vaccines and a lot of the benefits they have. Is there a reason scientists in the future are going to stay with more traditional vaccines? Is there a benefit to that?
I'll just bring up, when we sit here and we talk on our computers and we're texting and we're doing all this, we can think about how for us getting a vaccine, Jason already has his card. I'm jealous. But think about people who are in third world countries, for example. Think about people who are in Antarctica and they can't get over to the vaccine place, So not only getting there and getting injected and all this special treatment that we get, that works for us, but it doesn't always work. For example, in Africa. So, in Africa or some other population, wouldn't it be better if we had a pill? Wouldn't it be better if we had something that didn't have to be frozen at -80, etc. So I think, that's kind of a societal bent to that question, let alone the fact that, well, I'll just stop there.
I could answer a lot of the questions that are in there kind of all at once. There's a lot of this we don't know. And the reason it has to be stored at that, because they knew it was fragile. They didn't want to take a chance that they would get through the studies and have it be a waste of time if they didn't store it cold enough or didn't take certain precautions. So a lot of it, even with the second follow-up with the booster shots on some and maybe not on others, we just don't know. So they're taking, again, the best science, figuring out how they think they can best head off this pandemic, and then going with it. And now we sit around and wait to see, do we have to get one next year? Do we have to get one in 5 years? Do we have to get one next month? We don't know.
I'd add to what Jason said too, that when they do these approvals where they say it's got to be at -80 and you can only have it at 4 degrees for X number of hours or disaster happens, all that that's saying is that's a sufficient condition for it to work. And I guarantee you that right now they're looking at whether we can push the envelope a little bit. And it's possible that you'll see 4C delivery for these. Someone asked a question, why does the vaccine have to be stored so cold? mRNA can exist in our cells at body temperature. And it's a good point. The reason that we're kind of going with the cold storage is because we know RNA is a relatively kind of fragile transient molecule, but these are packaged into these lipid particles, and it's possible that they're more stable than we might have initially imagined. And so people are asking these questions right now. And just like Jason mentioned about, are we going to need annual boosters or something like this? We're going to have answers for these in the coming months. This just was, with warp speed, it was kind of, how do we get where we need to be as quickly as possible? And -80 for one vaccine and -20 for the other was kind of part of that.
So I think what I'm hearing is that essentially the approach is the abundance of caution approach.
Yep.
They want to make sure that it's not going to fail because too much is at stake.
Right.
Okay. So in terms of, I know there's some in terms of talking about the public, how, and some people talking about, you guys talking to the public may not be that useful because people don't necessarily believe scientists. But I mean, how do we talk to the public about this? How do we talk to them about to people who are leery about things? And I'm trying to translate a few of the audience questions and group some of them at this point because there are a lot of really good questions in there.
I could start off with that and probably tack a couple of them. The first thing when you're talking to somebody, as we've seen recently, it doesn't do us any good to try and beat somebody over the head to change their mind on something. So if you get somebody who thinks that, as it says in the question, that it's a hoax and it's an infringement on my liberties, if you really want to make a difference, ask them why they feel that way. Because sometimes and be sincere about it. Don't you want to know why they feel that way? And by asking them to explain why they feel a certain way, they have to go through it in their mind how they came to that conclusion. And that's a very, very powerful thing, because they sometimes will, after they go through it with you, and maybe they don't obviously change their mind at that point, but you counter and say, I understand what you're saying, and I appreciate that. I guess my point is this, but I respect your thoughts, and I hope that we can all come together on this. Give them credit for their thought process, because if we just sit there and point at each other and say, you're wrong, no, you're wrong, I'm wrong, we're not going to get anywhere. So that's part of it. And the other thing is ask them what is worrying. So after they tell you what's going on, why they think a certain way, try to talk to them about how to address those concerns. Some of the things that we talked about here. If they think the virus is a hoax, I don't know how you could possibly think that, but you don't say that. You say, well, I'm sorry that you think that way, but these people are dying from something. And I think that what I'm hearing is that this is something I can do to help reduce the chance of that spreading and hurting more people.
Not to fall into the same trap, but there's good research that shows that the approach you are suggesting works better.
Yeah, and I adding to what Jason said, I guess I would gently disagree maybe with the idea that scientists aren't the best people to communicate this. And there are places like Pew that do surveys of, they ask the public, do you trust X? And scientists typically max this out. I pulled it up as we were talking, and so the 2019 survey, it's 86% of the public has a great deal or fair amount of confidence. If you look at the numbers for other professions, it's not nearly as good. Religious leaders is 60%, the media is about 50, business leaders is 50, elected officials is like 35. People
Lawyers?
trust scientists.
So is that on the list, lawyers? I was just curious.
I did not have lawyers. The K-12 principals did pretty well, though. They were about 80%.
That's great.
Yeah, no, and I see what you're saying is that, if there's a level of trust there and somebody who's informed, who better to maybe present some of the information.
And what we want to do in all of this is not only to get them to open up their minds, but by them opening their minds and maybe changing it, they then become an ambassador that can pass that on. And, we talked about the warp speed, but creating a vaccine that's effective in and of itself does nothing. There needs to be an effective distribution system and a means of getting that into the arm of the patient. And as Kathleen mentioned previously, accessibility is a huge obstacle right now, not only with disparities in our Black and Brown communities, but in our rural communities and in some of the people in these pockets that don't have access. So we really need to look at that, not only to improve with this situation, but so we have that system in place for any future problems that we have.
I feel like we could probably go all night with this, but I know that we've passed our time here, and I don't want to take up everybody's time, even though it's clear that the audience is interested and passionate about this. But I do want to be respectful of our panelists' time as well. And I want to thank all of our panelists, all three of our panelists were fantastic. And thank you so much for taking the time to come here and speak with us about this. Really appreciate it.
Thanks, guys. This is a lot of fun.
Thank you, everybody.
Thank you
Enjoyed
for all the
the
Thanks
questions.
everybody for, thank you everyone for being here. And hopefully you'll tune in next time with us. Look forward to talking to you.
It was really fun. I'll tune in. See you in the Petri dish again.
Excellent.
I'll be there.
Thank
We'll
you.
hopefully see you in March. March 17th is our next Petri dish.
The questions were incredible, so thank you for coming to play with us.
Yeah, thanks
I
a
really
lot
have
to the
not
audience.
seen as many questions on any Petri dish, so
Record.
we kept me busy.
Really fun.
Bye, everyone.
Good
Thanks,
vaccination.
everybody.
Thank you.